Hello everybody, today we're doing a video on PCR, which is the polymerase chain reaction. I've actually been working in the lab recently and I did a PCR, so I'd like to explain what it is. So first of all, it is a elaborate technique that's widely used, and it allows scientists to make millions to billions of copies of a specific DNA like fragment or segment from even like a very tiny sample. So you can have one tiny sample and you can get a large amount of the specific DNA segment that you're looking for. So yeah, it also can be known as molecular photocopier. I mean. So yeah, and then before I start, I also want to tell you, like, the person who made PCR, his name is Carl Mullis, and it's very, like, it's a very interesting guy, not very good guy, but the way he figured out how to make PCR is pretty interesting, and I'll have a link below for an article about that. So first, before you do PCR, what are the things you need? So first thing you need is a DNA template. So for me, for example, I work with plants, so then I would need to get let's just say a leaf from the plant. So this leaf, and then I would have to take a chunk out of it, so then I can get some DNA. So, and then with that DNA, I can use that to get a specific. If I'm looking for a specific segment, I can use a PCR. But first, we need just some DNA to begin with. Secondly, we need an enzyme, DNA polymerase. This is what it's important. So for DNA polymerase, it's an enzyme that used to synthesize new strands of DNA. So let's say you have a strand here, some different colors, green, purple, and purple one. So yeah, so you have strand here, and then your DNA polymerase is basically moving from the 5' to 3' direction, just adding adding base pairs and making making the new DNA strand. So that's what DNA polymerase does. In the lab, we use one called Taq polymerase, which is commonly used because it's from extremophiles, which has a very high like point to where it melts. So like even because when you do PCR, you use pretty high temperatures. So if you have an enzyme that can resist those temperatures, that makes it much easier, much more efficient. The third thing you need is a primer. So primers are short synthetic pieces of single-stranded DNA. So they're usually 18 to 25 nucleotides long. And then the primer is basically your start position, and that's where you're trying to get to. So for example, let's just say, and the reason why it's 18 to 25 nucleotides is, if you think about it, if let's just say your primer was only 2 pieces, it was an A and a T. Oh, let's see, let's just say A and C, whatever. And then you have a long DNA sequence, which is like extremely long. Then if you, anytime there's a TG, that primer will attach to you. So it's not very good primer. So then that's why you would want one with like longer 18 to 25 nucleotides, because each position can have 4. So then it'll do whatever that's times the power 4. So then there's very low chance that that it'll attach twice. And then basically a primer, the same thing with transcription. When you're learning about that, is like that's like the starting position when you have your primer. That's where then the DNA polymerase will go from. And it's also important to know you'll have a forward and reverse primer, because when you have the DNA, it breaks to 2 strands. One will be down, one will be up. So yeah, so if you have, let's say our primer wants to attach to here. Then our primer will be up here attached to it with A, C, A, C from the 5. And then it'll attach here, and then the DNA polymerase will come on, start adding more base pairs to it. But that's like the starting position. So next thing we need is deoxynucleotide triphosphate. So that's your DNTPS. So that's basically just like a bunch of base pairs you can think of it, but it'll be like the full base pair. So then you'll have your glucose with your nitrogenous base with your phosphate. So yeah, so you have a bunch of you need a bunch of DNTPs roaming around. So then your polymerase, that's like the way this. So if this next letter was an A, and if this was a T, for example, then that's what will attach the DNA polymerase will attach it onto it. So you need a bunch of basically base pairs, or you can think of nucleotides roaming around. And then lastly, what you need is a buffer. Basically, what a buffer does is it provides an optimal chemical environment. So this allows for everything to run smoothly and allows everything to happen. So PCR is carried out in a machine called a thermocycler, so which rapidly changes the temperature of the reaction mixture. So there's 3 steps that are needed for this. I'll just do it on a different page. So we'll call these the 3 steps for PCR. So the first step is a denaturation step. So this will have very high temperatures that are required for this. So sorry. Let's say you have your sample and the buffer of your leaf. So that's all your DNA that you got doing other experiments. So if you zoom in, when the temperature rises, your double-stranded DNA, which looks, I'll just draw it like this, like connected like this, will end up looking like this. So from your double-stranded DNA turns into single-stranded DNA. And then you may be thinking, oh, how come they split so perfectly in the middle? That's because the backbone, so then the backbone of it, this part, different colors circular. This part is made out of phosphates, and it's like the diagram from the last one, like this, phosphates and sugar, and they're covalently attached. So that covalent bond is much stronger than the hydrogen bonds that connect between two strands. So these hydrogen bonds connecting them are not very strong, like especially compared to the covalent ones. So then that's why they end up breaking apart. So that's why you're left with single-stranded DNA when you raise the temperature. So yeah, so they melt. You can think of it like that, or separate into 2 single strands. So the second step is called annealing step. So this is the complete opposite. This is where you lower the temperature. It's still pretty hot, but it's much lower than what we started off with. Because like this is the first step is around, let's just say 94 to 98 degrees Celsius, and then this step is 50 degrees Celsius to 65 degrees. So it's still pretty hot, but there's much lower. So this step is allows for, let's see, so now you have your two single-stranded DNA, one up here, and let's say you have one down here, floating in your buffer. So, and then this one, let's say, is 3' to 5'. This one would be 3' to 5'. So now that they're apart and then it's cooler, this allows the primer. This allows the primer to attach. So let's just say for our example, to make it easy, we'll have a 4 base pair primer. So now the primer will attach in the forward direction. So it's 5' to 3'. That's what is easier. That's when you think about transcription. That's the leading strand. So then the forward primer will attach, and then when the temperature is lowered, and then the reverse one will also attach. So let's just say this one, which is a CCCAT. So now the other primer, I'll make it blue, will also attach. And then it's important to know that there is 2 of them. So yeah, that will attach to this one, but in the 5', 3' direction. But then it looks like the 3' to 5' direction. So now, because the temperature is lowered, it allowed the primers to bind or anneal to their complementary sequences. And it's really important that this step is not too high or too low, or else it wouldn't allow the primer to attach properly. And the last step is the elongation step. So now this one's pretty simple. So now the temperature is raised so that we can say for the Taq polymerase, it's 72 degrees. So that is the optimal working temperature for the DNA polymerase. So then with the first strand, I'll just use it. We can just completely copy this. So now with the first strand, well, I'll just use one because it's the same thing, but it will be more chopped up because it's in the reverse direction. But then the polymerase will always go from the 5'. So this is the Taq polymerase. It'll always move from the 5' to 3' directions. And as you remember, we have all these floating base pairs around that we added. Just everywhere floating around, and then that's what is picked up from the polymerase, and then it'll start elongating it. And then it'll continue adding the DNTPs to the DNA. So as you realize by this now, like if you think about it, in one cycle, so if you start off with one strand of if you started off with one, let's say one double-stranded DNA, in one cycle, we'll end up with 2 strands of double-stranded DNA. And then in another cycle, we'll end up with 4 strands. And So this is after 2 cycles. So and when you realize the pattern is 2 to the n, so after if an n is equal to the number of cycles, and so then 2 to the n is going to equal the amount of double-stranded DNA that you have. So, for example, they could be around, if you do an experiment and there's 20 cycles, let me get my calculator out. So that's going to become 1,048,576 strands of double-stranded DNA. So that's like a crazy amount. And I always like to think this is the way like Carl Mullis describes it. This is how he figured it out. So he imagined if he was on the, so let's say there's the Earth and there's the moon. If you're on the moon and you're trying to figure out one license plate from the moon, that's like impossible. That's like when you're trying to figure out the one sequence of DNA using a microscope, or you're trying to figure it out with just one, if you have only one strand of DNA, double-stranded DNA. So that's almost impossible to find. But if you have, let's say, the man on the moon, it says, if the whole world is covered in license plate, like every inch of the world of the Earth was covered, then it'd be so easy to find. That's the same thing with PCR. If you have over 1 million copies of your specific sequence or segment that you're looking for, then it's gonna be so easy for you to figure it out, to find the specific segment of DNA. And that's basically all that I have to say. Hope you guys learned a lot, and you guys enjoyed watching about PCR. Thank you guys for watching.